right, why don't we get started? Thank you all so much for joining us today for our next iteration of the public webinar series for the Common Fund data ecosystem. And we're going to start off with some updates and then we'll go into our featured speaker. But first off, I just want to remind everyone that if you're unable to attend any of our previous webinar series, that we have everything recorded and available on our YouTube. So please feel free to scan the QR code here for the playlist so you can take a look at those videos as well. This is going to be recorded and will be shared. So if there's any colleagues that are unable to join, please feel free to share this recording with them as well. And as always, if you have any feedback, questions, would like to be added to our listserv, please email us at help@cfdeknowledge.org. Here is our upcoming 2026 webinar schedule. If your program is interested in presenting on July 31st or November 20th, please let me know as we're still looking for individuals to participate, but we look forward to this incredible lineup from here on out. Here are some upcoming conferences where the CFDE members will be featured. This is only for the first half of the year. So as you can see, there's the U.S. Association for the Study of Pain in March, the American Physiology Summit in April, and BioIT World in May. For BioIT World, the training center will be hosting their 7th annual BioIT Fair Data Hackathon. This will be in Boston, Mass, on May 18th and 19th, with a focus on using open source tools and adhering to fair data principles. If you are interested in applying to be a part of this hackathon, there's some details on both the Knowledge Center and the Training Center's websites. And if you have any questions in particular, please feel free to shoot me an email as well. We're really looking forward to this session. As a reminder, we also have the Decoding the Data Ecosystem podcast, and it's more than just a podcast. It's really just a place for a community to be passionate about omics research and ways to solve complex biological puzzles and addressing pressing health challenges. This podcast is for basically anyone, whether you're a seasoned researcher, a student just starting out, or you're curious about the future of biology. This will have some insights and inspiring stories from community members. So please feel free to check this out as well. And then just one other event that we'd like to highlight, the Common Fund Data Ecosystem is going to host an incubator for Pan-NIH resources July 9th and 10th at the Neuroscience Center in Bethesda. And so essentially this will be, an event to share as a forum for NIH partners to identify shared goals, gaps, and collaborative opportunities. If you're interested in learning more about this, please feel free to scan the QR code. And again, email me if you have any questions. We are currently gauging interest of people that would like to participate, speaker opportunities, and there'll also be a poster session. So we have a little bit of details on that. Since this is in July, we are building the agenda out. So we'll share more information about the agenda and speakers as we get closer to July. And so with that, those are all my updates. I will hand this now over to Elizabeth Chun, who's going to be sharing somatic mosaicism across human tissues, the data analysis, and data portal. So thank you, Elizabeth, for joining us today. We're really excited to have you.
Yeah, well, thank you so much for having me, Mackenzie, and everybody at the CFDE. I'm happy to introduce to you our SMaHT network and also the data that we've been generating and how we've been using it to infer more knowledge about the human biology in terms of somatic variation. So very happy to be here, and I'll share my screen now.
And just a reminder to everyone, please feel free to use the chat or Q&A to ask questions. I will be monitoring those as Elizabeth is presenting.
Great. So yeah, my name is Elizabeth, and I'm a project manager of the Data Analysis Center here at Harvard Medical School in Dr. Peter Park's lab. so that everybody is on the same page in terms of somatic mutation and mosaicism, that is the key focus of our network. I just wanted to quickly just go through the background first before I delve into our network, to introduce you to our network and describe the data. And so somatic mutation basically are the mutations that we do not inherit from our parents, and these accumulate over time as we develop and age and be subject to various exposures or just the random and very natural biological processes that would involve DNA replication and error fixing. And so there are several sources for the mutations to rise as we age and develop. So mechanisms could include DNA replication errors during the synthesis as cells divide, and the lack of or inability for this replication errors to be fixed. Other source could be the direct DNA damaging agents such as UV light or smoking or drinking, etc., that would introduce these mutations. Other things are just spontaneous chemical reactions. So for example, thiamine could actually just spontaneously deaminase into cytidine. And so that would also cause mutations as a natural mechanism as well. And so, but then in addition to this natural mechanism, there are some biological susceptibility. And so there are some regions in our genome where that could be more prone to these errors to occur, or there are some genetic predispositions that could allow some mechanistic failure to occur more easily in some individual versus the other individual. And so all these mechanisms of mutations can be inferred when we look at what we call mutational signatures. And these are trinucleotide blocks of channels, what we call basically these changes that are occurring in the base, and we look at the flanking bases left and right, and together we are trying to infer based on these patterns of this different type of mutation occurring at different frequencies, we can derive unique signatures that can sort of infer what the underlying mechanisms that would give rise to the kind of signature-like mutational pattern that we see when we analyze sequence data. So we have some known well-characterized signatures that are linked to specific mechanisms such as MMR deficiency or UV light, smoking, etc. And also signatures that are associated with just aging as well, because as we age, we would accumulate more mutations. So these mutations we're treating as a footprint, because what we can imagine is that, I mean, we are all born, our life begin in a single cell, this fertilized egg, where these will start to divide. And every time cells divide, they have a chance of accumulating mutation. And these mutations are basically the footprint of the lineage of developed organs. And so tracing back these footprints, we can actually understand things about our development, how the branching of the cells as they develop over the lineage traces that gives rise to specific cell types. And so we can understand better about the biology of the differentiated cell types in terms of organ development. So many people have studied these for quite some time now. And what we are trying to show here is that what we also understand in terms of pathology is that I think cancer is one of the hallmark disease of the somatic mutation, where the disease is driven by the somatic mutation. And what we have learned over time is that these mutations occur way earlier in a person's life before a person develops cancer. And so understanding these dynamics of the clones that have somatic mutation and whether these specific mutations can give rise to clonal expansion, which really means is that this mutation somehow has conferred a phenotype or ability for cells to proliferate a lot faster than nearby cells, for example. So taking over the population with the specific clones with a specific mutation. And if by certain chance, if this one is the cancer driver, a mutation, then this could be a precursor to a malignant development later on. And so there has been many people who are clinically involved in understanding this somatic, what we're calling mosaicism. So this term mosaicism came up where if you can look at cell as a, that's shown here in circle in different color, indicating cells with different kind of mutation, you can actually look at our body or our organs, our cellular population as a mosaic of cells with different genomes, so to speak, where this difference come from this mutations arising as we develop, which are the somatic mutations. And clinically, this somatic mosaicism has a quite significant relevance because only 20 to 50% of the patients are diagnosed using exome-based sequencing to try to find mutations in the coding regions of the genome, which is quite very little, as you know, less than 5%, which means that a lot of these mutations that are responsible for giving rise to many systemic disease are found in the non-coding regions, or these are complex genomics where a single variant cannot really tell you what overall underpinning of these pathologies. And that these variants are hard to detect because they are not found systematically. There could be also routinely for these genetic testing, the blood is taken, but if the mutations that that are cause of these diseases are only found in a subpopulation of cells or affecting parts of the body, then it's important that we actually use that specific tissue to understand better about the genetic underpinnings of this disease. And so understanding this mosaicism is critical, and people have found in the clinical setting that mutations that was not found in blood was actually found in a formaldehyde fixed, paraffin fixed samples that were later taken and excised for regions of suspected cells that might be responsible for certain blood vessel related disease as shown here. And then that's where they found the mutation not in blood. And so understanding the landscape of these somatic mutation in the normal cells or normal tissue is of key importance. And this detection of mutations that are found in a subclonal population is quite difficult because, as you can see, if mutation is found in all cells in our body, such as germline mutation or mutations that are found very early on in our development stage, then we don't need a lot of, I guess, signal to tease apart from the noise. However, if the mutations that are only affecting subpopulations of cells, for example, allele frequency of less than 10%, then we need way more, I guess, stronger power to detect these rare events. And how we usually detect these mosaic variants are quite simply, it's a function of coverage where let's say if every position in the genome that you see on the right side is sequenced about 100 times, and let's say about 6.5% of that read contains the evidence for a variant that's found in this position, we are basically translating that to the allele frequency, and we can infer the cells, if this is heterozygous mutation, then we can infer that about 12.5% of the cell fraction in the tissue that I've just sequenced contains this mutation. And so these are important metrics as we try to understand the characteristics of this variant and to infer the cellular lineage. And as you can infer, the mutations that occur earlier up in the earlier stage of the development would have a higher variant allele frequencies. The ones that are found occur really late in the in development will only affect a few cells and therefore be present as a mutation with a much lower frequency, allele frequencies. So as we are trying to push the limit of detection to detect these rare mutations that may arise later in life, there are many challenges that we face bioinformatically. So in which all can give rise to this false positive signal. And so it could be that the sequencing error could give rise to us calling the mutation as a putative somatic mutation. If a DNA sample, let's say, gets contaminated with another human being, let's say somebody coughed during sequencing experiment, and somebody's germline mutation could be viewed as my somatic mutation, things like that. And also, if the reads have mapped to the wrong region, this misalignment give misinformation about where the variants are, where it wasn't a variant. In fact, it was just the wrong read mapping to the wrong place of the genome, and the mismapping of the region was the false positive. So there are various things to overcome. And of course, this is a large endeavor to solve this important and complex problem. And so basically the somatic mosaicism across human tissues network, in short, SMaHT, is consisting of multiple research centers around the country. And largely our network is consisting of the 5 different components, where organization center is at WashU, which is responsible for coordinating the network activities and doing the outreach, also liaise with the NIH and other programs, such as other programs here at the CFDE. The tissues that we sequence or profile are procured at the NDRI, which is designated as a tissue procurement center, or in short TPC, and they are responsible for collecting and distributing tissues from the donors that we profile in the network. And so at the SMaHT network, we are targeting up to 15 or 19 tissues from postmortem donors. And by year 5, we are aiming to profile 150 postmortem donors. We then have 5 different centers who are very well-established sequencing centers around the country, namely Baylor, Broad, New York Genome Center, University of Washington, and Washington University, that are designated as the Genome Characterization Center. And these are the centers that will have a production level throughput to use the state-of-the-art genomic sequencing technologies to apply what we are calling core assays, which I will explain a bit further later. We are the Data Analysis Center, or in short, DAC, and we're the one who's getting all the data that's generated from the network members, and we are processing, integrating, and analyzing them, and build a portal through which we can distribute the data in the right secure access, tiered access system. And we are also responsible for building, in collaboration with all the network members, building the variant catalog, which will be basically the backbone of the somatic landscape, so to speak. What is the profile of the somatic mutations that are seen across different tissue types in the normal individuals, or phenotypically normal individuals. And lastly, in order to develop, although we are using the state-of-the-art technologies, we are also aiming to develop next generation computational and experimental tools to further improve the detection and analysis of these somatic mutations. And so these are tasked to what we are calling technology and tool development projects. And these are consisting of the 14 different research groups throughout the country. So the network goal is to identify and characterize the somatic mosaicism in normal human tissues. And to do that, we use multiple different technologies, such as duplex sequencing, which has the error rate even below the sequencing error, so that this is actually geared towards identifying very rare mutations that would traditionally be very difficult to identify with a very high accuracy. Another one is to use the single cell method. And so things like PTA or DLP plus are the whole genome sequencing of the single cell level to infer the cell lineage. What are the mutations that are occurring at a single cell level? On top of that, several, such as University of Washington and our group as well, we're devoted to build a reference genome that is personalized. So what we are calling donor specific. So right now, the common practice is to align the sequenced reads from one individual or a specific sample to what we are calling reference human genome, which are basically an aggregate of multiple individuals that was drawn from different type, mostly from blood, but other different tissues. And it was basically a reference is it's not an individual specific genome. And so if there are any individual specific variations, that it's very hard to detect which one is actually just a misalignment or one it's actually an individual variation that is not mapping correctly in when it was mapped to the reference genome. So in order to improve the accuracy of such calls or variant detection, we are trying to assemble a donor-specific reference genome and then use that to increase the variant accuracy as well. And because we are generating multiple tissues from the same donors, we are trying to integrate that across or see what are the tissue-specific differences. And also using this multimodal sequencing data types, we are trying to also use that to either orthologously validate the call or to understand better the advantage and disadvantage of each of these different sequencing technologies in terms of calling variants. And so through all that, we are trying to improve the current state of the computational and analytical methods of identifying somatic mutation. So far, our project, which has entered its third year, actually nearly finishing third year. We started in May 2023. And the first data was submitted quite quickly, within 6 months' time, the same year. And our data portal that we built was built and was ready for event live, ready with the functionalities for data downloads in following year in February. So we were working pretty hard to make this network hitting the ground running. And we started with what we call benchmarking phase, where we used cell lines or tissue homogenates that wouldn't have spatial or biological heterogeneity when we send out the samples to different sequencing centers, so that we can focus on things that are truly technological differences to be able to fairly compare the outcome of profiling a biological sample. And so using this cell line or tissue homogenates, we performed benchmarking experiments. And it was twofold aim, where we wanted to generate the biological samples that was ideal for benchmarking for other community use. So these cell cultures or cell lines or tissue homogenates are available for people who wants to use this for their own experiments and compare with our results, for example. And also second aim was to do the benchmarking of the computational methods to identify what is the optimal methodological paradigm where we could identify rare variants or low BAF, let's say BAF less than 2%, to identify these mutations. And so we were comparing different technologies, different protocols, computational tool parameters, etc., benchmarking 11 methods with about 7 groups that are participating in what we called a variant detection challenge that went on in the second year. Starting last year in January, we started to actually go into production mode and generating data from the intact tissues from the postmortem production donors. And the goal was to comprehensively profile each donors. For that, we are generating about 150x. It's 2 sequencing centers that are generating 150x Illumina, altogether 300x Illumina whole genome sequencing data for long read on PacBio or ONT platform, we're generating whole genome sequencing data between 60 to 120x. On top of this whole genome sequencing, we also are generating the bulk total RNA sequencing data for each of these tissues as well, depth of up to 150 million read pairs. And in order to supplement this, what we are calling the core assays, we are also generating the Kinnex data, which is the mRNA sequencing on PacBio, so that we can actually capture the complete full-length transcripts. And in addition, we have the duplex sequencing, other transposable element targeting sequencing to detect very specific types of the structural variants arising from transposable elements. And we are generating data and assembling genomes that are donor-specific. And so currently, we completed sequencing 25 donors that was generated from about 1,500 tissue samples across about 19 different tissue types. And we are aiming to complete more donors by the end of year 3, which is in 2 months. Okay, so I think I explained the core assays to you, which is shown on this table on the left. So altogether, we generated about a little over 350 terabytes of raw sequencing data, which we all analyzed and aligned and made it released to the data portal for data sharing across the network members. And it's about 300 terabytes as well. And these are the list of the novel assays that are applied from and developed by the tools and technology development groups, some single cell data sets, and we focus a lot on duplex sequencing. So we apply 6 different types of duplex seq methods. The L1, the retrotransposable elements that we are targeting for these sequences, and L1 derived sequences and sequence onto different platform to identify this mobile element mediated insertions. And we have some other sequencing technologies that are geared towards understanding better the epigenetic characteristics of these tissues, especially how it actually relates to the somatic mutations that might be found in a particular sample. Okay, so just an example of what we have made available. So the Colo829, for example, that we used for benchmarking experiment is open data. So if you come to our data portal today and have go through self-registration, you will be able to download these data sets. And so you can see that these are quite high short read sequencing data, and as well long read as well. And so these are some of the examples of the depth of sequencing that our network has produced over time. Okay, so at the Data Analysis Center, what we have done was to do the data processing, and we also apply quite a number of QC checks as well to ensure the high quality of the sequencing. Also checking for any sample swap or sample duplicate possibilities, and if there is any other different human sample contaminating this particular sample, or if there is any bacterial or other microbial contamination that might be happening. So through it all, after the QCing, we make these aligned and processed BAM file available for the analysts to then call the mutations. And we are also working with them closely to implement this standard, gold standard pipeline that is designed to produce the what we are calling the reference variant call sets. So I'm going to go into some of the analysis that within the Data Analysis Center have performed. And so, as I explained before, we were engaged quite intensely in the benchmarking exercise, trying to identify what might be the best paradigm or best computational workflow to detect rare variants using benchmarking samples. And we have identified that the use of the Illumina short read call mutations at the beginning with a high sensitivity that are later validated using long read to achieve high precision, like would be the best practice going forward. And so based on this work, we started to apply this, we built the pipeline and started to apply this practice to detect mutation in the production donor. And this is the work by Corinne and Hu in our lab, for the postdocs in the Park lab, have basically working on analyzing the whole genome Illumina data and validating with the long read to detect these mosaic somatic mutations, and basically finding interesting things like on the x-axis are the different tissue types, on the y-axis is the number of mutations, and the individuals with heavy smoking obviously have a higher number of mutations that are found, and then in the liver with the cirrhosis, for example, in certain individuals have a higher number of mutations. And we are also happily seeing the consistent finding of the age association with the somatic mutations, that older people having more mutations, that we were also able to observe in our own sample using our own methods as well, as a way to validate our approach. And we are continuing to, with the network as a whole, we are trying to infer biological relevance of underlying all these mutations, somatic mutations that we are seeing in different types of tissues. And so we are doing things like, comparison of a different, across mutation burdens across different tissue types. And we are also trying to understand what the extent of the spatial heterogeneity in the intact tissue. And so with the TPC, basically each sequencing center is getting different, what we are calling cores. So these are these frozen samples. The sections of the samples are taken in form of a tissue core, and being able to see what mutations are unique in one core versus another as a way to understand the extent of the spatial heterogeneity has been quite interesting and important investigation. So that's shown on the x-axis here to show what is the core-to-core variation. Okay. Okay. I went pretty fast, but I also want to sort of take a breather here, at least to see if there is any questions from the chat, or am I good to continue?
You're good to continue at the moment, but this is a great reminder. If you have any questions, please feel free to raise your hand, use the chat or Q&A. And if not, we can continue and we'll have other questions at the end.
Okay, great. Yeah, thank you, Kenzie. So I wanted to sort of pause here also to sort of regroup and kind of think about what it means to, detect mutation in terms of biology, and what technology allows us to go, basically what will allow us to see things in more depth. And being able to accurately detect rare mutations will allow us to look at deeper and deeper into cell population, from just at a tissue level down to cell population level, down to group of cells, down to a single cell. And here is a really helpful plot that was made by one of my colleagues here, Vinay, in our lab. Basically on the x-axis in the top, it's showing different kind of technologies or data types that we have. Like, so normally when people want to detect germline mutation, they will say, we need to have a 30x whole genome. And when we are quite serious about detecting somatic mutation with high accuracy, then the coverage of the whole genome starts to really climb to rather commonly used is 200x for quite serious studies. And then and people can have a single cell, a panel of single cell, up to about 500 cells are sequenced at a time. And then there could be some targeted sequencing can be done. And then the single cell, single nuclear level. And with these different data types or technology or methodological approaches, it allows us to see cell population at different depths. And so obviously the ones that are low depth bulk whole genome sequencing will allow us to detect the cells, mutations that are found at frequency of 50% that is found in all cells. And going down to right now, we are at a resolution that we can detect a single cell in 100 million or billion cells at a time. And that technology is achieved by single cell whole genome sequencing. And so being able to accurately read out these rare mutations will allow us to know biology at a single cell level. Oops. So one of the technology that has the very high accuracy is the duplex sequencing that we are using a lot. On the left side, we are trying to show you like basic concept of what the duplex sequencing is, which basically is being able to sequence the single strand in independent sequencing so that the mutations that are found in both strands, we are certain that this is a true mutation call. And with this level of accuracy achieved on the right side, we are able to detect the mutation that occur at a frequency much lower at 100, 1 to the 10 to the minus 7 or 8, being able to basically accurately call something that's even lower, even more accurate than surpassing the error rates that we see in the typical Illumina platform. Another endeavor that Vinay is undertaking is identifying mutations that are in a non-coding region. So many functionally important somatic mutations are actually occurring in a non-coding regions where these regions are open in terms of the chromatin accessibility, allowing for proteins such as transcription factors and other enhancing proteins to bind to and modulate gene expression or other epigenetic changes. And so one of the techniques that allows us to sequence the regions that are open, more accessible for other protein to bind and modulate its function is called ATAC-seq. And so he's using ATAC-seq data to call the mutations or somatic mutations that are occurring in these open regions that might be more functionally related. And so he and Chris Walsh lab, who are one of the TTD groups in the SMaHT network, are developing this Somatic technology to enable accurate detection and analysis of the mutations that are occurring in the open access, open chromatin regions using ATAC-seq data. Other endeavors in our lab is to detect somatic structural variants. And this is a work that's largely undertaken by the postdoc in our lab, UA, in collaboration with Adam English and Fritz Sedlacek's group at the Baylor College of Medicine. And so in this benchmarking experiment, she was using cell culture that was mixed with 6 different normal cell lines, which what we are calling the HapMap individuals. And so using this synthetic benchmark samples, she was basically comparing different methods and different data types that allows us to detect somatic structural variants with high accuracy. And so the kind of different types of structural variants that we could detect are, of course, insertions and deletions, as well as those that are occurring at a very low BAF, things that are, and she was also able to, UA was also able to characterize what might be the factors that would hinder with accurate structural variant calling, such as presence of tandem repeats, or whether there was any different certain characteristics that are in the flanking regions of the SB events that could also give rise to the false positive calls. So through this exercise, she was able to derive set of recommendations or best practices for the community. And all these work that I am showing you in our presentation are in the preprint, if not published. And these are, together with the network members, we are preparing a publication package that is summarizing this benchmarking exercises that we have performed as the network. So going back to the best practice in terms of the somatic structural variants, on the top, plots that you're showing here is the allele frequencies, structural variants that are occurring at different allele frequencies, from super rare, below 0.5% on the left, all the way going to, I guess, relatively more common variant types occurring at allele frequencies less than 20% on the right. And on these three panels are the type of data that was used to call structural variants using Illumina, PacBio, HiFi, or ONT sequencing. And the metric on the y-axis is the recall, the precision, and the x-axis is a different coverage, sequencing coverage. And one of the key take-home message from here, for example, is that Illumina remains very cost-effective for detecting variants that are found in higher frequencies. Whereas long reads are essential. So for example, if you look at the bottom plot, for example, the fact that the lines are just linearly increasing with the increased coverage means that the precision of the somatic structural variant discovery is dependent on the coverage of the long read, and that it's very important to have a basically impact of the high coverage long read availability in being able to detect structural variant is quite highlighted in her work. So going forward, another effort that our group is doing is to find a better way to construct telomere to near complete whole genome assembly, haplotype resolved telomere to telomere assembly, using the data that's relatively more readily available and in relatively more cost-effective way. And so one of the ways that people usually do the genome assembly is to first use PacBio data for high accuracy, and then in order to fill the gaps, try to make use out of the ultra-long ONT data, which is quite expensive, and then use the things like Hi-C data for phasing so that we have haplotype resolved whole genome assembly. But in Flora and Hung and Hao Yu were able to develop this new method called Hi-Phism ONT, where it's only using the regular ONT data. No need for PacBio or ultra-long ONT or Hi-C for phasing. And so what she has done, and she and Hung and Hao Yu have done, was to compare with the standard practice, which is to use Furco as the assembler and Haro as the ONT data, error correction, and compare the performance in terms of the runtime or CPU usage, as well as the quality of the assembly that was the resultant of using these two different approaches and compare them. And basically the key take-home message was that using only the standard ONT, using this Hi-Phism ONT tool, we were able to derive quite accurate and continuous telomere to telomere genome assembly, using a lot fewer CPUs and no need for using GPUs, for example, and then run it about 10 times faster. And so this is now published, and this is one of the ways that we are going to use in order to assemble tissue-specific genome assemblies or see ways to sort of alleviate the cost of generating this personalized genome assemblies within the SMaHT network. So this is just to show you the quality of the genome assembly using these HiFiSim ONT method compared to the gold standard Verco plus Harrow methods at the bottom. Just to tell you that this is the region in the genome where it's very notorious to assemble accurately because of very high sequence homology, basically revolving in a gene locus, SMN 1 and 2. Because of the high sequence homology, it was hard to sort of accurately assemble it through. And using HG002 T2T Q100 reference, the gold standard reference genome, we were basically comparing the genomes that are assembled using HiFiSim versus Verco and Harrow to show you that the whole block of green on the top, which is the assembled genome using HiFiSim, showing that this was assembled correctly compared to the gold standard, whereas the current the method was actually struggling a little bit using only the ONT data, where it did okay. Actually, it did perfectly with ultra long. So if people are sort of resource conscious about generating ultra long ONT, there is a tool now available to just be able to accurately construct the whole genome using the ONT data only. So I want to sort of conclude my talk with going through the portal a little bit so that you can start become familiar with the SMaHT data portal and what kind of data that we host and the future feature that we are planning to include in our data portal. And as the DAC, of course, we are managing and we are overseeing and implementing the analysis pipelines that would be universally applied to all the SMaHT data, and also working closely with the working groups to incorporate these novel development or new tools that we find helpful to increase the accuracy of the variant calls so that we can make these somatic variant catalog available for the community to download and be able to distribute and visualize and be able to explore this data through the data portal, which is the repository and analysis hub for the consortium and hopefully for the greater the research communities. And I just wanted to tell you that our portal is built on well-established open source infrastructure that runs on AWS. And the tools that we have made that forms the backbone of the data portal is all from these published work that has been generated from our lab before. All this is to say is that this is a very high functioning, scalable data portal that allows us to automate metadata validation and submission, data processing, curation, analysis, release. And now we're in a process of designating our portal as the NIH controlled access data repository as well. And the tiered access level, being able to interface with the dbGaP for the different types of users is also one of the future feature that we're going to be working hard to implement. And so the URL for our SMaHT data portal is here. Right now, the data is accessible to the network members only. But as I said before, the benchmark data from COLO829 or HapMap, which are open access data, it requires one to self-register and then be able to have access to these open access data immediately. And so if you have any questions about the data access and about the portal or a feedback of its look and feel, please free to contact us at the help desk that's shown on the homepage of our data portal. It's shown on the orange box here. Just to show you the kind of data visualization, because this is a lot of different types of data, this is one of the visualization aid to kind of show you as a fun, you can move this around. It's quite interactive, just to appreciate the complexity of the data that we're generating for the SMaHT network and what that might be used in order to identify different types of somatic variants. Other features that we're looking to incorporate are things like what we are called things like Chromoscope, which is developed by one of our staff scientists here in the Park Lab, Dominic. So this is a way to visualize different types of mutations, including structural variations in a what we are calling the circles fashion, and then be able to see these by interactively where they are in different locus across the genome, and be able to have a read level view of different, I guess, evidence for different variant types, looking at the alignment. So here, for example, this is a location of the breakpoint of a deletion event, and that we detected in the sample that we analyzed, of seeing that there's basically a very, like a cliff where the reads are soft clipped and not aligning to certain regions, and the depth of sequencing is basically quite low, indicating that there's a deletion events going on here. So the plan going forward for us is that, of course, we are working hard to make this available for wider scientific communities to come to the data portal and access the data. And the protected data is going to be under dbGaP control. And so I encourage people who are coming to our data portal, even if you are only intending now to get access to open data, please register using your institutional email address that is linked to login.gov or ERA Commons, so that when we turn our data portal to public facing fully, integrating to work as the controlled access data repository, that your access continues to be able to access the protected data using dbGaP authorization authentication. So you basically use the email that you're going to be registering at the dbGaP when you apply for the approval, so that you don't have to do anything. You don't have to create yet another account or have a problem accessing the protected access data in our portal. So to finish off, the portal is built on this excellent team of people at the DAC. So our PI is Peter, wonderful PI who's spearheading our data analysis center. And we have the backend, front-end data modeling, curation, and bioinformatics development team who are just fabulous people to work with. And I'm very fortunate to have wonderful team members such as these people. So with that, I'd like to conclude and make time for Q&A if there are additional ones.
Thank you so much, Elizabeth. That was a fantastic presentation. Just a reminder, you can ask questions via the chat, Q&A, or if you want to raise your hand, we can take you off of mute and you can ask directly as well. Just give it another minute or so.
Sure, yeah.
And if you have any questions that arise afterwards, you can also email Elizabeth, I'm sure. Okay, well, I think that must have been a extremely, oh, actually, there is a question. I was going to say you covered everything in that presentation. But anonymous attendee has said, why would these postmortem tissues require controlled access?
Oh, although this very generous donors have passed away, their genetic information, especially germline information, is still informative entities for people who are related to them. So usually the whole genome sequencing coming from an individual, a human subject, is almost always protected access for that reason.
Great, thank you.
Yeah, thank you for that question.
Okay, we'll just see if there's any others. It's very informative.
Well, thank you. I'm glad that you found it informative and for others as well.
Okay, well, I thank you for those who asked questions, and thank you so much to Elizabeth for presenting today. We're really thankful that you had been able to join us. Our next webinar will be March 27th, and we look forward to seeing you all there. We'll have some updates from the Knowledge Center. So thank you again. We greatly appreciate it. Bye, everyone.
Great.
Bye.
Bye.